Chapter 12, Section 1, RNA. Learning objectives. Describe how RNA differs from DNA. Explain how the cell makes RNA. When Watson and Crick solved the double helix structure of DNA, they realized that the structure itself did not explain how a gene works. Eventually, scientists learned that another nucleic acid, ribonucleic acid, or RNA, helped to put the genetic code into action. RNA, like DNA, is a nucleic acid that consists of a long chain of nucleotides. In a general way, genes contain coded DNA instructions that tell cells how to build proteins. The first step in decoding these genetic instructions is to copy part of the base sequence from DNA into RNA. RNA then uses these instructions to direct the production of proteins, which help to determine an organism's characteristics. Like DNA, RNA is made up of nucleotides. Each nucleotide consists of a 5-carbon sugar, a phosphate group, and a nitrogenous base. However, DNA and RNA differ in 3 important ways. Unlike DNA, RNA uses a sugar, ribose, instead of deoxyribose. RNA generally is single-stranded, and RNA contains uracil in place of thymine. These chemical differences make it easy for enzymes in the cell to tell DNA and RNA apart. RNA and DNA have completely different roles. DNA is a bit like an architecturalist master plan, and it has all the information needed to construct a building. But builders never bring the valuable master plan to the job site, where it might be damaged or lost. Instead, they work from blueprints, which are inexpensive, disposable copies of the master plan. In a cell, the DNA, or the master plan, remains safe in the cell's nucleus, where it serves as a template to make multiple RNA copies. Then those RNA, or blueprints, travel to the ribosomes, which then put the coded instructions into action by assembling proteins in the cytoplasm. You can think of an RNA molecule as a working copy of a gene, which is a functional segment of DNA. RNA has many roles, but for now we're just going to focus on one of their roles, which is protein synthesis. RNA controls the assembly of amino acids into proteins. There are 3 main types of RNA involved in protein synthesis: messenger RNA, or mRNA, ribosomal RNA, or rRNA, and transfer RNA, or tRNA. Like workers in a factory, each type of RNA molecule specializes in a different aspect of the job. The first type, mRNA, or messenger RNA, is the molecule that carries copies of these instructions from the nucleus to the ribosome in the cytoplasm. Most genes encode instructions for assembling amino acids into proteins. Therefore, this molecule is important for carrying all of the information to the cytoplasm. The second type, ribosomal RNA, are subunits. Proteins are assembled on a ribosome, which are small organelles composed of 2 subunits. Those subunits are ribosomal RNA and as many as 80 different proteins. During the assembly of a protein, a third type of RNA, tRNA, carries an amino acid to the ribosome and matches them to the coded mRNA message. A single DNA molecule may contain hundreds or even thousands of genes. However, only those genes being expressed are copied into rRNA and given at any given time. The process of copying a base sequence of DNA to RNA is known as transcription. Transcription is similar to DNA replication, but the product is an RNA molecule instead of a duplicate DNA. The transcription segments of DNA serve as templates to produce complementary RNA molecules. Transcription is carried out by an enzyme called RNA polymerase. RNA polymerase first binds the DNA and then separates the DNA strands. It then uses one strand of DNA as a template to assemble nucleotides into complementary strands of RNA. The ability to quickly copy a DNA sequence into RNA makes it possible for a single gene to produce hundreds or even thousands of RNA molecules. How does RNA polymerase know where to start and stop making a strand of RNA? The answer is the RNA polymerase does not bind to DNA just anywhere. The enzyme binds only to protomers, which are regions of DNA with specific base sequences that can bind to RNA polymerase. Other regions of DNA cause transcription to stop when an RNA molecule is completed. Like a writer's first draft, a new RNA molecule sometimes requires a bit of editing before they are ready to be read. These pre-mRNA molecules have bits and pieces cut out of them before they can go into action. The portions that are cut out and discarded are known as introns. In eukaryotes, introns are taken out of newly synthesized pre-mRNA molecules while they are still in the nucleus. The remaining pieces are known as exons. Exons are then spliced back together to form the final mRNA. What is the purpose of making large RNA molecules and then throwing parts of the molecule away? That's a good question, and biologists still do not have a complete answer. Some pre-mRNA molecules are cut and spliced into different ways in different tissues. Because of this, a single gene can actually produce several different mRNA molecules. Introns and exons may also play a role in evolution, making it possible for very small changes in the DNA sequence to have dramatic effects on how genes affect cellular function.